Okay, moving on to section 8, we're going to talk about the small intestine. And when we talk about the small intestine, this is probably our most important organ for our digestive system, because the small intestine is the major site for both digestion and for absorption. I'm not worried about the statistics, how long it is, or anything like that. I just want to say that from the pyloric sphincter, pyloric sphincter is the sphincter that's connecting the stomach to the duodenum, to the ileocecal valve, which is at the end of the small intestine, so the end of the ileum, that's going to connect to the large intestine. So those are kind of two valves that allow entry into the small intestine and exit out of the small intestine. As I mentioned previously, there's 3 subdivisions of your small intestines. Duodenum, which is closest to your stomach. The jejunum, which is going to be your middle segment. And then your final segment is going to be your ileum, which is ultimately going to connect to your large intestine. So as far as the blood supply goes, the superior mesenteric artery is going to bring most of the blood. We do have veins, so the veins are going to drain into the superior mesenteric veins, and then ultimately into the hepatic portal vein, and finally the liver. When we're talking about nerve supply, we have both innervation from the sympathetic and the parasympathetic nervous system. So this is just kind of showing you, here's our stomach right here. What's connecting our stomach to the first segment of our small intestine, or the duodenum, is the pyloric sphincter. That's where we're squeezing out about 3 milliliters of chyme. That's what it was going through. Once our stomach connects to the duodenum, the next segment in blue right here, which is pretty large, is going to be our jejunum. And then our final segment of the small intestine is the ileum, which is going to connect to the large intestine via the ileocecal valve. We do have some modifications in the small intestine as well that are going to help increase the surface area. Anytime we increase the surface area, its predominant function is to help increase either breakdown or absorption. So some of the modifications that help contribute to the increased surface area include circular folds. These are really deep, permanent folds of the mucosa and the submucosa. And the whole purpose of having these throughout the area is to help slow the movement of food and allow time for full nutrient absorption. Our next thing is the villi. And villi are just finger-like projections of the mucosa itself. What do? They have dense capillary beds and lymphatic capillary that we call lacteal. So these are going to help in absorption. Our third one is going to be microvilli. These are long, really densely packed cytoplasmic extensions of the cells of the mucosa. Kind of give it a fuzzy appearance, which you've probably heard mentioned to be the brush border before. And on the microvilli, there are enzymes that we refer to as brush border enzymes, which help with the final breakdown of our carbohydrates and the proteins in our small intestine. Okay, histology-wise, again, a lot of this is just really going to be for lab, but I'm just bringing up some of the important parts here. We do have modifications in the tunics of our small intestine as well. So we have modifications of our mucosa and submucosa. We have intestinal crypts. These are just tubular glands that are interspersed between the villi itself. We have 5 major types of cells that are found within the mucosal epithelium of the villi and of the crypts. So enterocytes are our first one, and they make up the majority of the epithelium. They're just simple columnar absorptive cells that have tons of microvilli on them. So function really to help absorb nutrients as well as electrolytes. And if we're talking about the crypts, their function is going to be to produce the intestinal juice, which is just a watery mixture that contains the mucus and helps serve as a carrier fluid for absorbing nutrients from chyme. So anytime we have simple columnar, we have goblet cells that are associated with that type of epithelial tissue. So we do have the presence of goblet cells, which are just mucus secreting cells. Our third type of cell is the enteroendocrine cells, and these are the source of the enterogastrones that we talked about earlier. So this is where our secretin and cholecystokinin are coming from. Paneth cells, these are found deeper in the crypts themselves, and they help by releasing antimicrobial agents, so things like defensins and lysozyme that are going to help destroy some bacteria, so we don't have huge bacterial colonization within our intestinal lumen. And our final cell are going to be stem cells, and these are the cells capable of undergoing mitosis and dividing. Sometimes they can differentiate and become other cell types. The next thing I want to talk about is the mucosa-associated lymphoid tissue, or MALT, and this includes both individual lymphoid follicles as well as aggregated lymphoid nodules. So the biggest thing here is the Peyer's patches. Peyer's patches are the aggregated lymphoid molecules, and they're located in the lamina propria. They're found in great numbers in the distal part of the small intestine. So Peyer's patches are found in the ileum. So histology, if you put in a section of the ileum, you'll be able to see these very large Peyer's patches there. Where we have these Peyer's patches in the ileum, that's where we're going to see a huge amount of bacteria. Because the lamina propria also contains huge amounts of plasma cells that secrete immunoglobulin A, or IgA, that's going to help protect against intestinal pathogens. The submucosa is going to consist of areolar tissue, not important for lecture, but just put on the slide. We also have duodenal glands in the submucosa of the duodenum that are going to be producing alkaline or bicarbonate-rich mucus, whose function is really just to help neutralize that acidic chyme that's coming in from the stomach. So intestinal juice. The intestinal glands secrete anywhere from 1 to 2 liters of intestinal juice every day. The major stimulus for the production of this juice is going to come from hypertonic or acidic chyme. Normally, intestinal juice is slightly alkaline, so anywhere from 7.4 to 7.5 as the pH, and it's going to be isotonic with the blood plasma. When we're talking about intestinal juice, it's predominantly water, but we also have some mucus, which is secreted both by the duodenal glands and those goblet cells that we were talking about earlier of the mucosa. So let's get on to the digestive processes in the small intestine. Remember, we picked up with chyme being kind of squirted in at 3 milliliters from the stomach. The chyme that's coming in has partially digested carbohydrates and from our salivary amylase, and partially digested proteins from our pepsin that was produced from our stomach, as well as undigested fats, or mostly undigested fats, because the lipases in our stomach weren't really contributing to the bulk of the lipid breakdown. Typically, it takes about 3 to 6 hours in our small intestines to absorb all of the nutrients and most of the water. So, sources of enzymes for digestion. We have bile, we have bicarbonate digestive enzymes, not just those brush border enzymes that we were talking about before. These are all going to be imported though. The small intestines isn't producing them themselves. They're being brought in from the liver and from the pancreas. Brush border enzymes that are bound to the plasma membrane do help perform the final digestion of chyme. So chyme entering the duodenum is usually hypertonic. Hypertonic because it has all the food particles that have begun to be broken down. But chyme delivery has to be slow, like we mentioned before, to help prevent osmotic loss of water from the blood. So we do have to take into consideration the osmolality of the contents that we're bringing into our duodenum relative to our blood. We also have acidic chyme that has to be adjusted upwards. So we don't want to keep that high acidity that's being brought in. We want to increase the pH of it. And the chyme has to be mixed with bile and pancreatic juice in order to continue digestion, because those are where our enzymes and things needed to continue breakdown are located. We also have our enterogastric reflex and our enterogastrones that are going to help control the movement of food into the duodenum itself. So just kind of talking about what happens after a meal. If we take a look at the small intestines and we have a lot of intestinal contents, we have what's called predominantly segmentation, which we mentioned in the first section, or is the main motion of the small intestines. Again, we don't have control over it, so it's going to be initiated by pacemaker cells. And basically the whole purpose of segmentation is to help mix and move the contents towards the ileocecal valve. Intensity is altered by long and short reflexes and those hormones that we talked about. The more intense the contractions, basically the greater the mixing. Between meals, we have peristalsis occurring. That's going to increase. Motilin is going to be the hormone that's responsible for helping increase peristalsis. Any meal remnants, bacteria or debris, those are going to be moved towards our large intestines. If we're talking about the complete trip through the small intestine from our duodenum to the ileum, it takes about 2 hours. So this is just giving you a table for the control. You can go in and take a look at this along with the reflex and the hormones that are involved in either increasing gastric motility or in reducing it.